On August 1st, 1920, African-American tobacco farmer Henrietta Lacks was born. She lived her life working on her family's tobacco farm in Virginia from sunup to sundown and caring for her children, David Jr., Lawrence, Elsie, Deborah, and Joe. As a black woman in the 1950s, she had minimal education and carried herself with only the knowledge necessary for everyday life. Not much is known of her personal life, but what little is known of Henrietta is compiled in a book titled The Immortal Life of Henrietta Lacks, and few pictures were ever taken of her. She later died of an aggressive form of cervical cancer that began as carcinoma in situ on October 4, 1951. Though she lived her life engaged in the domestic duties of her family's farm, she is immortalized in history for her unknowing medical and scientific advancements. Unbeknownst to Henrietta and her family, she helped develop medical miracles such as the polio vaccine, cancer treatment drugs, and even foundational landmarks for the COVID-19 vaccine. How? Henrietta Lacks is the woman behind the HeLa cell, and science stole her cells to create this HeLa cell line, which can regenerate forever and serve as a living, regenerating, bioengineerable being for scientific experiments and medical breakthroughs. The first immortal cell line. The harvesting of HeLa cells was highly unethical by today's standards. However, they emerged as a crucial turning point, enabling the creation of the polio vaccine and building a lasting impact until the COVID-19 pandemic of 2020, when they were used to develop the COVID-19 vaccine. On January 29, 1951, Henrietta Lacks went to the Johns Hopkins Hospital, one of the only hospitals accepting black patients, after feeling a hard lump in her cervix. They found that she had stage 1 malignant cervical cancer. There are two kinds of cervical cancer, one the doctors believed remained on the surface of the cervix, and one that had already invaded the surface of the cervix. World-renowned pioneer gynecologist Dr. Richard Talind and cell culture pioneer Dr. George Guy were working together to prove that non-invasive cervical cancer would eventually lead to invasive cervical cancer. In doing this, they took tissue samples from everyone they could get their hands on, which included Henrietta. Henrietta Lacks signed a consent form stating, "I hereby give consent to the staff of the Johns Hopkins Hospital to perform any operative procedures and under any anesthetic, either local or general, that may deem necessary in the proper surgical care and treatment of." Her consent form said nothing about post-operative procedures pertaining to cell cultures. They took her cells and shortened her name, giving the potential cell line the name HeLa. On February 5, 1951, she received her biopsy results. Just a few months after the operation, on October 4, 1951, Henrietta died. With the help of his wife Margaret, Dr. Guy had already attempted to create an immortal cell line for quite some time with no success. But immediately after HeLa entered his lab, it started to multiply, filling any space it was given. Once Guy realized HeLa was immortal, he gave it to his friends and colleagues, which gave anyone access to not only Henrietta's cells, but what was used to feed and grow the cells, a special mixture called a cell culture medium.
Dr. and Mrs. George Guy of the Johns Hopkins Hospital in Baltimore established this cell strain, which has been in continuous culture since 1951.
In the 1950s, poliovirus ran rampant, and scientists everywhere needed a way to prevent it. At the Tuskegee Institute, a historically black institution, Dr. Russell W. Brown and Dr. James H.M. Henderson led a team of researchers to develop a vaccine for polio using rhesus monkey cells to test on. The rhesus monkey cells were expensive to produce, and HeLa was discovered to be inexpensive and quick to manufacture. In the spring of 1953, just 2 years after Henrietta Lacks had died, the National Foundation for Infantile Paralysis funded a cell culture factory at the Tuskegee Institute, where the polio vaccine trials were being held. The doctors found out that her cells were highly susceptible to polio and could grow substantial amounts of it, making it optimal to begin testing.
Randy was the first child to receive an injection of salt polio vaccine in the field trials in 1954.
Less than 2 years after switching to HeLa, on April 12, 1955, the vaccine was developed and put into use. In September of 1957, HeLa cells were used again to test the harmful effects of X-rays. The test determined that human cells have less than a 40% chance of survival when faced with 96 Roentgen, a measurement of radiation, where a typical chest X-ray provides the equivalent of 1/1000th the previous measurement. Notably, HeLa cells have also been used to pioneer a cell testing method that tests if a cell line is cancerous. They have been sent to space to study the effects of zero gravity on human cells, and they have been used to study the effect of hydroxyurea on blood disorders like sickle cell anemia. In 1973, more than 20 years after her surgeon, Dr. Lawrence Wharton Jr., took a sample of her cervix, Henrietta's daughter-in-law Babette finally found out that doctors secretly kept part of her alive and used her for science. Because Dr. Guy had changed her name when he took the cells, there were no identifiers for her family to know who she was. And until her cells and their ability to reproduce rapidly started to contaminate other cell lines, the press and doctors using them had no interest in finding out who the woman behind HeLa was. At a conference in 1966, Stanley Gartler first dropped the news that HeLa could contaminate other cell lines. From this point on, doctors were finally interested in finding out who HeLa was, and they all began to decode her DNA and become increasingly interested in who the human being was. He later gave a lecture at Washington State's very own WSU Pullman campus.
There had been a contamination. People had let the HeLa cells, by sloppy techniques, grow into another thing. At that time, the people doing cell culture didn't really have much feeling for the field of genetics. They didn't think of using markers such as this to check whether their cell lines were true or not.
When Dr. Guy died on November 8, 1970, his colleagues wrote a tribute to him about his most commendable achievement, the HeLa cell line. They used her original biopsy notes containing her name and wrote, "Henrietta Lacks as HeLa, an immortality which has now reached 20 years. This article was the first time her name had appeared in a published text, but this was only a scientific journal for a small field. It was not until it was published 2 years later in a Nature article that it reached more ears than just the scientific community. It even landed itself in a Rolling Stones article in 1976. Scientists and doctors are still using HeLa cells in research today, as well as much of the research done with them. During the 80s, heavy research was done on cancer and cancer treatment using HeLa cells. Using HeLa cells, researchers were able to identify that HPV can lead to some cervical cancers, making significant strides against the kind of cancer that Henrietta herself had. Scientists also focused on cancer treatment drugs during the 80s, finding a drug that slowed the growth of cancerous HeLa cells called camptothecin. Because of this early research, we currently have over 150 approved cancer treatment drugs for 31 types of cancers. Additionally, HeLa cells advanced HIV studies significantly. Scientists found that HeLa cells were less susceptible to infection by HIV, and using this information, they found out how HIV infects people and are currently developing a preventative HIV vaccine. Through these HIV studies, researchers discovered that Ebola infects the body in a comparable way, and in 2019, the FDA approved the Ebola vaccine, providing the first step in eradicating a virus that has been wrecking Africa for nearly 50 years. Henrietta's case also brought about a question of patient privacy, and it is her case, and a few others, that were the cause for the Health Insurance Portability and Accountability Act, also known as HIPAA. When Rolling Stone published her name in an article, it put her family at the center of attention without their permission. The court system decided patient privacy had to be protected, meaning their names could not be released alongside their personal information, under a pseudonym or otherwise. After 92% of the human genome was completed on April 14, 2003, a new generation of DNA sequencing technology gave people the resources to know all information regarding a person's genetics when giving their DNA. This issue led to concerns that people would get discriminated against for their susceptibility to certain conditions. Thus, the Genetic Information Non-Discrimination Act, or GINA, was passed as an extension of HIPAA. Most recently, HeLa cells were used in creating the COVID-19 vaccine. The realization that HeLa cells were not easily infected by COVID led to biological engineering. The cells lacked a specific receptor called the ACE2 receptor that allowed the virus to enter the body. The goal of this was to find the individual gene to target with the vaccine, and on December 11, 2020, the COVID-19 vaccine was approved for those 16 and over.
And the FDA has given its full and final approval.
Henrietta Lacks is an unsung hero that changed medicine forever. Her contributions will be forever cherished and appreciated. However, because the harvesting of her cells and their journey to immortality was done in secret, her family has gotten no compensation until just last year. Thermo Fisher, a company that makes an annual revenue of over $40 billion, a decent proportion coming from the manufacture and distribution of HeLa cells, has only recently come to a settlement with the family of Henrietta Lacks. Her story brings forth questions of great moral debate. Nonetheless, thank you, Henrietta, for providing a turning point in medicine and patient protection as we know it.